In this video, we will examine the structure and function of enzymes. Enzymes are biological catalysts. Catalysts are substances that accelerate the rate of a reaction by tunneling the activation energy. They do this by increasing the number of molecules in the 3D configuration that is needed to complete the reaction. One way of explaining this is the lock and key theory. In the lock and key theory, the enzyme is thought to have a specific three-dimensional shape, which includes an active site. Normally, reactants in a reaction must collide with enough energy and in exactly the right position to form the proper bond. In the lock and key theory, enzymes remove the reactant from random motion and rearrange the shape of the reactants so that they can form a new intermediate known as the enzyme substrate intermediate and complete the reaction in the proper positions. Here you see one reactant can only fit into the active site of the enzyme in one way, and in that way that reactant is held in exactly the right position so that the other reactant can only come in one way. They are put together. This would be the enzyme substrate intermediate. The new product is released, and the enzyme is free to carry out another reaction. In this case, the enzyme known as sucrase. Most enzymes end with the ending ase, and this tells us that this enzyme carries out the reaction involved with the sugar sucrose. So we see the formation of sucrose by the enzyme sucrase, or we can also see the breakdown of sucrose into its monosaccharides, glucose, and fructose. An active site. Into that active site, the substrate sucrose can fit. When they are in the active site, they are held in the perfect position to allow water to come in the hydrolysis, and then the two glucose and fructose molecules are released. If the enzyme is present and there is a large amount of glucose and fructose present, the sucrase will do the reverse reaction. They will enter the active site, the water will be taken out, and the sucrose will be released. So we have the reverse reaction occur. We will see later that this is what happens in cells when cells develop what are called organelles. Organelles are compartments that control what enzymes are exposed to, meaning if the enzyme is exposed to glucose and fructose, sucrase will make sucrose. If the enzyme is exposed to sucrose, the enzyme will catalyze the reverse reaction. A modification of the lock and key hypothesis to explain enzyme actions is what is known as the induced fit hypothesis. And here what we see is the active site represented by A does not exactly fit the substrates B1 and B2. However, when B1 and B2 enter into the active site closes in around it. The enzyme molds itself to accommodate the substrate. In induced fit, the active site does not have a very rigid shape. At this point, we can talk about two different types of enzymes. An absolute enzyme would be more indicative of one that has the rigid active site, like this one. An absolute enzyme has a highly specific active site that can only catalyze one substrate. Another type of enzyme, better explained by the induced fit hypothesis, would be group-specific enzyme. So this is an enzyme that is specific for a group or moiety found as part as a molecule, and it does not have to match the whole molecule to catalyze the reaction. This image represents what is known as an apoenzyme. An apoenzyme is an enzyme that is actually a conjugated protein. You should remember that conjugated proteins are proteins that require a non-protein group or a prosthetic group to complete its active site. Here, the prosthetic group is called a cofactor or a coenzyme, and what it does is it completes the active site for the apoenzyme. Most cofactors or coenzymes are vitamins, such as vitamin B2 or riboflavin. which is necessary for the formation of FAD. Vitamin B3 or nicotinamide is necessary for the formation of NAD and NADP, and vitamin B5 or pantothenic acid is responsible or necessary for the proper formation of coenzyme A. All of these apoenzymes, the three we just named, are used in the energy generation reactions of respiration and photosynthesis. One other explanation or another representation of the apoenzyme, here we have the protein portion of the enzyme, which is inactive. It does not have an active site. The cofactor joins it, and now it has the active site, which allows the reactants to fit and carry out the required reaction. Competitive inhibition will slow down the reaction rate of an enzyme-mediated chemical reaction. Competitive inhibition is very simply explained by lock and key or induced fit hypothesis. Another molecule has a similar shape to one of the reactants, and it competes to occupy the active site. Once it comes into the active site, the inhibitor does not allow the true reactant or its second reactant to fit into the active site, and basically what it does is it stops the reaction or slows the reaction by occupying the active site. It is considered competitive inhibition because this molecule, the inhibitor, and this molecule, the normal reactant, are competing to occupy the site. So that if we increase the concentration of the reactant that we want, it will compete and eventually it will replace more of the inhibitors, as we will have less inhibitor to occupy the active site and more of the reactants to occupy the active site. So the substrates cannot get in when the inhibitor is present. In non-competitive inhibition, we are working with what is called an allosteric enzyme. And an allosteric or regulatory enzyme has two active sites. And in non-competitive inhibition, the allosteric site, or the second active site, is occupied by the inhibitor, and when the inhibitor occupies that site, it permanently alters the active site so that the reactants do not have access to it. If we increase the concentration of the reactants, they still will not be able to displace the non-competitive inhibitor. So the reaction rate will be permanently altered. Now, when we look at allosteric enzymes, what we've got is two possibilities. You can have allosteric inhibition, where the inhibitor occupies a second site, and when it's in that allosteric site, it distorts the active site and stops the reaction. There is a second type of allosteric enzyme where we can have an allosteric activation, where to begin with, the reaction is off because the active site does not accept the substrates. An allosteric activator occupies the allosteric active site, altering the true active site so that it can now accommodate a substrate. There are 5 different factors that affect reactions that are catalyzed by enzymes. Those factors are temperature, pH, substrate concentration, the presence of inhibitors, and the presence of cofactors. And these 3 graphs represent how those factors affect an enzyme. Most enzymes have, in terms of temperature, what is called an optimum temperature for them to function, so that below that temperature the rate of the reaction will be slower. Above that temperature the rate of the reaction will be slower. And enzymes have evolved to work at temperatures that the organisms possessing those enzymes need them to function at. pH works in the same way. Enzymes have an optimum pH. Below that pH, the rate of reaction will be slow. Above that pH, the rate of reaction will change. The last graph represents how the concentration of a substrate affects the rate of reaction and enzyme activity, and you see as more and more substrate is present, the rate of the reaction increases until at this point all of the enzymes are occupied, and increasing the substrate does not increase the rate of the reaction, and we reach a plateau. So the rate of reaction remains constant. Here we see four graphs that represent four situations in relation to cofactors and inhibitors. The first graph is the effect we just looked at as how the rate of reaction changes as the substrate concentration changes. At this point, we've got all of the enzymes' active sites occupied by substrate so that the reaction does not increase, it reaches a plateau. If we have a competitive inhibitor, you can see that as the inhibitor is present, it slows the reaction. This is the curve with the inhibitor present, but as we increase the concentration of the substrate, you can see that we do eventually displace the inhibitor, and we do at some point reach what is like the normal curve for concentration of the substrate. When we have a cofactor, you see that the presence of the cofactor increases the rate, and when we have a non-competitor inhibitor, here with the non-competitive inhibitor, you see that the reaction never changes or reaches what would be the normal reaction without the inhibitor. So you can see that the rate of the reaction is slowed and reaches a lower plateau because a large number of the enzymes are permanently occupied by that inhibitor and not available no matter how much more substrate we add to the system. The explanation of how temperature and pH affect the enzyme is the idea that high temperature or high pHs, the normal protein becomes denatured, which means that the hydrogen bonds or interactive forces between the carbonyl groups and the amino groups in the backbone of the protein, and the forces of attraction between the residue or side chains are affected by temperature and pH. And what that does is breaks down those interactions, which causes the protein to denature or completely change its shape. And when it changes its shape, it has no function. This is one of the reasons why we are very concerned when you have a child and the temperature goes above 103, because that would be the point at which human enzymes begin to be denatured. So what you would always do is try to cool down the individual so that that does not occur.